Today, we seek to explore an overview of genetics and inheritance. At the end of this lecture, you should be able to explain what is meant by genetics and inheritance, how the study of genetics, for example, started, the extent to which it has progressed, the importance of genetics, and so on and so forth. You are warmly welcome to another session of a Daniel Hayford's lecture. To begin with, note that the field of genetics itself is a branch of one of the fields of science called biology. It refers to the study of genes, genetic variation, and heredity in all living organisms. Did you know some authorities also define genetics as the discipline of the field of biology which explores thematic areas such as the function and physical nature of the genetic material, as well as inheritance and variation. Furthermore, genetics can be defined as the branch of biology which is concerned with the study of inheritance, including the interplay of genes, DNA variation, and their interactions with environmental factors. What kind of organisms do geneticists study? In the broader sense, the living organisms which are studied by the geneticists encompass those which have prokaryotic cells and are therefore known as prokaryotic organisms, and those which have eukaryotic cells and are therefore referred to as being eukaryotic organisms. Once upon a time, all prokaryotes were classified under the kingdom Monera by the scientific community. Today, prokaryotic organisms are celebrating the membership of two of the three domains of life, namely archaea and bacteria. The domain archaea is also called archaebacteria, and the domain bacteria is called eubacteria. The word archaebacteria could be interpreted to mean an ancient form of bacteria, whereas the word eubacteria could mean bacteria in a true sense. The other domain of life is called eukarya. Members of the domain eukarya, which the geneticists study, are the kingdoms Animalia, Plantae, Fungi, and Protista. Apart from living organisms, geneticists also do study those acellular organisms which are said to be at the edge of life, namely the viruses. Dear friend of Daniel Hayford, what about the acellular disease causal agents called virusoids, viroids, and prions? Do geneticists study such agents too? Please conduct some research and present your findings as comments on this lecture on the YouTube channel dubbed DH Media GH. Ladies and gentlemen, the human being, the domesticated plants, fungi, and animals of the human being, pests or vectors which spread diseases to the human being, and the pathogens which cause diseases in human beings are part of the living things which are explored in the field of genetics. So, why do you think the study of genetics is important to you, your family, community, country, continent, and the world at large? According to authorities in the field of genetics, knowledge and understanding of the various genetic factors genetic diseases or disorders, and their relationship with environmental factors, for example, are key in planning, implementing, monitoring, and learning more about promoting public, animal, and plant health and preventing diseases in proactive ways. Among human beings, for example, various genetic modifications or changes in the bodies of people of one generation, okay, have been associated with an increased risk of having subsequent generations with defects or developmental disabilities or developing economically important diseases such as diabetes, sickle cell disorders, cancers, as well as heart and blood diseases, among other things. I repeat, among human beings, for example, various genetic modifications or changes in the bodies of people of one generation have been associated with an increased risk of having subsequent generations with birth defects or developmental disabilities or developing economically important diseases such as diabetes, sickle cell disorders, cancers, as well as heart and blood diseases, among others. The field of genetics is truly important to you as an individual or a member of a family due to the fact that it can help you to explain the following with certainty. 1. What makes you unique or one of a kind? Do you remember that even the good book says in Psalm 139 verse 14 that you are fearfully and wonderfully made, or you are fearfully distinguished? Yes, genetics will help you know that you are fearfully distinguished. Yes, genetics will help you to explain why members of your family look alike, or why certain medical conditions like the sickle cell disease, cancers, and diabetes run in families, and how knowledge about your family health or medical history can help you stay healthy and or avoid or delay onset of certain conditions you might have inherited. Again, genetics will get you enlightened enough to know why you should not only know about your family health history, but also why you should share such information with your health care service provider. Dear friends of Daniel Hayford, genetics helps us to know and understand how living organisms work and how they can be improved to help meet the needs of humanity. Knowledge and understanding of the genetics of animals, plants, and mushrooms, for example, can get our farmers and breeders well informed to make informed decisions about what to raise and how to raise them. In addition, the concepts, principles, and techniques in the field of genetics can be employed to develop novel varieties of animals, mushrooms, and plants which are more resistant to drought, diseases, floods, and pests. Those which have short generation time, comparatively higher yields, or are better matched to certain specific growing and developing conditions. Furthermore, knowledge and understanding of the field of genetics can be used to formulate or, if you like, create consumable animals, fungi, and plants with desired characteristic traits such as nutrition, flavor, and taste. If you become a geneticist, you can afford to help us to improve the quality of the food we eat, the materials we use to produce drugs and clothes, and the effectiveness of our agricultural systems. Over the years, the field of genetics has truly advanced considerably along and all through various principal pathways. Such key or major pathways include experimental breeding activities, which started through the celebrated works of one Gregor Mendel, an Australian monk who had a background in science and mathematics. Through his efforts and the effort of many other scientists, it is now a commonplace that genetically diverse lines of organisms can be crossed to yield diverse combination of alleles in one line. Parental lines, for example, are crossed to produce first filial or F1 generation. The members of the F1 generation are then made to undergo random mating to yield pure breed offspring, progeny, or descendants with genotype, say, capital T, small t, small w, or capital W, capital W. This then now becomes the source of new plants, fungi, and animal lines and are useful as laboratory stocks for basic scientific or specifically genetic research works. This experimental breeding tool, technique, strategy, or procedure can also be applied in the area of commerce to produce the contemporary novel transgenic commercial lines called genetically modified organisms, GMOs. Indeed, many authorities have stated that many of the animal, fungi, algae, and plant species or varieties which are on the market today are consumed by human beings are bred through this technique. I repeat, indeed, many authorities have stated that many of the animal, fungi, algal, and plant species or varieties which are on the market today and are consumed by human beings are bred through this technique, which is the genetic engineering technique. Such food resources include bulls, cows, goats, tomatoes, maize, mushroom, pig, potatoes, rice, sheep, tilapia, and wheat. Another key pathway through which the field of genetics has advanced is known as pedigree analysis. Pedigree analysis was designed primarily to assist us to gain a better knowledge and hence understanding of the inheritance of genes from ancestors or parents to bring their offspring, children, or progenies. I repeat, pedigree analysis was designed primarily to assist us to gain a better knowledge and Hence, understanding of the inheritance of genes from ancestors or parents to their offspring, children, or progenies. It was designed as a chart which shows a family tree with the family members and their traits respectively. Although the pedigree analysis was originally developed to study the inheritance of genes in human beings, it is also valuable when exploring any population of living organisms when progeny data from some of their generations is limited. The strategy is also valuable when exploring species of living organisms which have a long generation time. By generation time, we mean the average interval between the birth of an individual and the birth of its offspring, or the average time between two consecutive generations in the lineage of a population. In nature, such a generation time usually ranges from minutes to hours, days, weeks, or years. Ladies and gentlemen, in a nutshell, a pedigree is a chart which diagrams the inheritance of a trait or health condition through generations of a family. It particularly shows the relationships among family members and or when the information is available, indicate which individuals have a trait of interest. The third pathway which we seek to explore is population genetics. Dear friends, population genetics is an interesting subfield of genetics which explores the genetic composition of biologically relevant populations and the changes or modifications in genetic composition which are attributable to the operations of a variety of factors such as the famous natural selection. Experts in this subfield of genetics do design abstract mathematical models of gene frequency dynamics, extract predictions with special reference to the probable patterns of variation in rare populations, and test the hypotheses, ideas, or educated guesses against pragmatic, real-time, practical, or empirical information or data as generated through research. Ladies and gentlemen, cytogenetics is another pathway through which genetics has progressed. Did you know cytogenetics refers to the study of chromosomes, which are long strands of DNA and protein which contain most of the genetic information in a cell? The cytogeneticists perform a laboratory test on cell, tissue, blood, or bone marrow samples, for example, to try to find changes in chromosomes or chromosomal aberrations such as broken, extra, missing, or rearranged chromosomes. Did you know changes in some chromosomes may be a sign of some types of cancer or a genetic disorder, disease, abnormality, or some other conditions? The techniques of the subfield cytogenetics could be employed to help health workers to diagnose a genetic disease, disorder, or condition, plan the treatment of such a disease, disorder, or condition, or determine effectiveness of treatments. The next pathway of the field of genetics is called molecular genetics. This subfield of genetics deals with the structure and function of genes at the molecular level and embraces the techniques of genetic engineering, which refers to the direct manipulation of a living organism's genome. Ladies and gentlemen, epigenetics is at times dubbed epigenomics. It is a field of study in the field of genetics which focuses on changes in DNA that do not involve alterations to the underlying sequence. The DNA letters and the proteins that interact with DNA can have chemical modifications that change the degrees to which genes are turned on or off. Certain epigenetic modifications may be passed on from parent cell to daughter cell during cell division or from one generation to the next. The collection of all epigenetic changes in a genome is called an epigenome. Genetic engineering, ladies and gentlemen, is the direct manipulation of DNA to alter an organism's characteristic traits in a specific manner. And it is another pathway through which genetics has progressed. It can also be defined as the process of altering the genome of a living organism. It ranges from altering or changing a single DNA base to deleting or inserting an entire region of DNA. Forget not the fact that molecular genetics typically deals with the molecular basis of inheritance and variation of genotypes and explores the link between the information-bearing macromolecules called DNA and RNA and the proteins which they encode. Did you know? All the applied branches or subfields of genetics depend on genetic principles and are used in agriculture, biotechnology, pharmacy, and medicine. Ladies and gentlemen, what do you think constitutes a gene? A gene is a unit of heredity existing as alleles on the chromosome, and that in diploid organisms, characteristically, two alleles are inherited with one of the two coming from each parent. Or a gene can also be defined as a discrete unit of hereditary information composed of a specific nucleotide sequence in DNA or RNA in certain types of viruses. Or, in simple terms, a gene can be defined as a sequence of nucleotides in DNA or RNA which encodes the synthesis of a gene product, and that product can be either RNA or protein. Note that genes of humans are situated in 8 chromosomal DNA and a few occur in the DNA of mitochondria. What about genetic variation? Ladies and gentlemen, the term genetic variation refers to the difference in DNA among individuals. It can also be defined as the presence of differences in sequence of genes between individual organisms of a species, or it is the differences among the genomes of the members of the same species. It enables natural selection, which is one of the primary forces driving the evolution of life to prevail. Ladies and gentlemen, for the purpose of emphasis, recall the fact that genes are units of hereditary information and that they carry instructions for building proteins. It is the genes which are encoded within such proteins which enable cells to function. Most sexually reproducing organisms have two copies of each gene, since each parent cell or organism contributes a single copy of its genes to its offspring. Note also that genes can afford to exist in slightly different forms called alleles, and that further add to genetic variation. I repeat, note also that genes can afford to exist in slightly different forms called alleles, and that situation adds to um, genetic variation. Ladies and gentlemen, did you know it is the combination of alleles of a gene which an individual receives from both parents which actually determines the genotype for a specific trait like skin color, height, hair texture, etc. The genotype which a person has for a specific trait in turn determines the phenotype. And the word phenotype refers to the observable characteristics including brown eye, green eye, blue eye, black skin, black hair, as well as curly, straight, and wavy hair. Genetic variation can originate from mutations, that is, the changes in the sequences of genes in DNA. The variation can also result from gene flow, that is, the movement of genes between diverse groups of living things. To conclude, variation may be due to sexual reproduction as it results in the creation of new combination of genes. Variation makes it possible for some living things to survive. Variation makes it possible for some living things to survive better than others in the environment in which they live. Living organisms of a small population can still vary greatly in their adaptation to a certain environment. Moths of the same species but whose wings have different color, for example, are a good illustration for this. Moth species with the color of their wings being similar to the color of the bark of the tree camouflage better than those of a different color. I repeat, moth species with the color of their wings being similar to the color of the bark of the tree camouflage better than those of a different color. So, the tree-colored moths tend to be better at surviving, reproducing, and passing on their genes. This is natural selection, the main force which drives evolution. Ladies and gentlemen, friends of Daniel Hayford, I just mentioned the term genome. What is a genome? A genome is all the hereditary information. That is to say that a genome is all the genes of an organism. The human genome, for example, contains somewhere between 20 and 25,000 genes. Hereditary refers to the process of passing on of traits from parent to their offspring through either the sexual reproduction or asexual reproduction method, leading to the offspring cells or the living things acquiring the genetic information of their parents. As indicated, variations between individuals can accumulate through heredity and cause species to evolve through natural selection. I repeat, variations between individuals can accumulate through heredity and cause species to evolve through natural selection. Note that one can define genetics to mean the study of heredity in the field of biology, and that heredity is also referred to as inheritance or biological inheritance. I repeat, one can define genetics to mean the study of heredity in the field of biology, and that heredity is also referred to as inheritance or biological inheritance. It was one Gregor Mendel who discovered certain key laws of heredity after conducting carefully executed experiments with garden peas during the middle of the 1800s. Mendel's laws spell out how genes are inherited from generation to generation in a dominant and recessive manner in accordance with the laws of probability. Mendel employed test crosses to show that the genes are like particles that are passed from generation to generation. His laws apply to all sexually reproducing organisms, be they plants or animals, including humans. Yes, including you and I. So they apply to the inheritance of many human genetic disorders. Today, a family pedigree can be used to reveal the mode of inheritance of a human trait or disorder. I beg your pardon. Mendelian genetics also helps us to know and understand inheritance by multiple alleles, degree of dominance, and the ability of some genes to have more than one effect. Many human characteristic traits are controlled by more than one pair of genes, and the resulting phenotype is influenced by the environment. Genes can be carried on both the autosomes and on the sex chromosomes. X-linked disorders are controlled by the genes on the X chromosomes. Because males inherit a single X, they always express a recessive allele located on these chromosomes. Because males inherit a single X chromosome, they always express a recessive allele located on this chromosome. Ladies and gentlemen, dear friends of Daniel Hayford, the popular concept during Mendel's time and before he conducted his experiments and formulated his theories is called blending theories of inheritance. This theory states that parents of contrasting appearance produce offspring of intermediate appearance. Mendel's findings were in contrast with this popular opinion. He formulated the particulate theory of inheritance, which states that inheritance involves reshuffling of genes from generation to generation. How? Gregor Mendel conducted genetics research through garden pea species as his experimental object. Unlike the preceding plant breeders, his research work employed non-blending characteristics of the garden pea. Mendel applied mathematics, employed the scientific method very meticulously, and kept careful records of his experiments, including the methodology and the data. So he established particular. I beg your pardon. So he established the particulate theory of inheritance. He established the particulate theory of inheritance, and thereby successfully invalidating the blending theory of inheritance. Furthermore, he crossed heterozygous plants with other heterozygous plants and noted that the recessive phenotype recurred in approximately half of the F2 plants, and that there was a 3:1 phenotypic ratio. Due to this result, he proposed his law of segregation, which states that the individual has two factors for each trait, and the factors segregate into the gametes. His law of segregation states that the individual has two factors for each trait, and the factors segregate into gametes. Ladies and gentlemen, Mendel did two test crosses. In which the F1 individuals displayed both dominant traits, albeit there were 4 phenotypes among the F2 offspring. The phenotypic ratio was 9:3:3:1. So he deduced the law of independent assortment. The law of independent assortment states that the members of one pair of factors separate independently of those of another pair. Accordingly, all possible combinations of parental factors can take place in the gamete. He used the laws of probability to compute the anticipated phenotypic ratio of a cross. A large number of offspring is counted to observe the anticipated outcomes and to be sure that all probable types of sperm have fertilized all probable sorts of eggs as is performed in a Punnett square. I repeat, a large number of offspring is counted to observe the anticipated outcomes and to be sure that all probable types of sperms have fertilized all probable sorts of eggs, as is performed in a Punnett square. The Punnett square. Ladies and gentlemen, the Punnett square makes use of the product law of probability to establish possible genotypes among offsprings. Afterwards, the sum law is used to establish the phenotypic ratio. Again, he crossed the F1 plant, having the dominant phenotype with homozygous recessive plants. The 1:1 ratio result he observed showed that the recessive factor existed in these F1 plants. That is, that they were heterozygous. Today, this is called a test cross. The reason is that it is employed to test whether an individual displaying the dominant trait is homozygous dominant or heterozygous. The two-trait test cross approach makes it possible for us to establish whether an individual organism displaying two dominant traits is homozygous dominant for both traits or for one trait only, or heterozygous for both traits. Many human traits and genetic disorders can be elucidated via simple Mendelian inheritance. Geneticists usually construct pedigrees when they are studying human genetic disorders to reveal the pattern of inheritance of a trait within a family. The actual pattern shows the way a trait is inherited. Pedigrees can be constructed for autosomal recessive and autosomal dominant patterns. There are other patterns of inheritance besides Mendel's contribution. Some genes, for example, possess multiple alleles, albeit each individual possesses just two alleles, as in the case of the inheritance of blood type in humans. Inheritance of blood category also shows codominance. With incomplete dominance, the members of the F1 generation are intermediate between the parent phenotypes. Of course, this occurrence does not support the blending theory in the sense that the parent phenotypes recur in F2. In the case of incomplete penetrance, certain characteristic features which are dominant may not be expressed due to reasons which are not known. In pleiotropy, one gene has multiple effects. This situation is seen in the case of Marfan syndrome and sickle cell disease, for example. Polygenic characters are regulated by various genes which confer an addictive influence on the phenotype, giving rise to quantitative variations. A bell-shaped curve is observed due to the fact that various environmental influences lead to various intervening phenotypes. An example of this is seen in the case of the inheritance of height in human beings. Other examples of multifactorial inheritance, that is, a kind of inheritance brought about by polygenes and the environment, are skin color and eye color. In Drosophila species, as in the case of human beings, the sex chromosomes are the determinant of the 1. The sex of the individual organism, with XX being female and XY combination being male. Experimental support for the chromosome theory of inheritance was birthed when Morgan and his colleagues reported that the gene for a characteristic feature which is not related to sex determination in any way, namely the white eye allele in Drosophila species, is located on the X chromosome. Alleles which are located on the X chromosome are referred to as being X-linked. Alleles which are located on the X chromosomes are referred to as being X-linked. Accordingly, whenever the geneticists are handling X-linked genetic problems, it is their tradition to designate the sexes through the use of sex chromosomes and to designate alleles using superscripts attached to the X. The Y chromosome is left blank because it does not carry these genes. Color blindness, adrenoleukodystrophy, Menkes syndrome, and hemophilia are X-linked recessive disorders in human beings. Recall the fact that it was through a good experimental design and other things that Gregor Mendel was able to establish his laws of genetic inheritance. Did you know his laws are applicable to human beings in many ways, despite the fact that humans do not often give birth to a large number of offspring? Sound historical records of genetic inheritance in families, including Mormon families, made it possible for scientists to reveal that several human genetic disorders are truly regulated by a single allelic pair. Such disorders include methemoglobinemia, cystic fibrosis, osteogenesis imperfecta, hereditary spherocytosis, and Marfan syndrome. Mendel probably succeeded because he studied a living organism called the garden pea, whose observable characteristic features are usually controlled by a single allelic pair. This observation is important because observable traits in living organisms are controlled by several genes or are influenced by additional factors, including environmental factors. Such other categories of genetic inheritance patterns are considerably different from the simple Mendelian inheritance. Indeed, the research works which were carried out by Morgan and other scientists did demonstrate clearly that the sex chromosomes contain genes which are not related to the gender of the living organisms which are bearing those sex chromosomes. I repeat, the research works which were carried out by Morgan and other scientists did demonstrate clearly that the sex chromosomes contain genes which are not related to the gender of the living organisms which are bearing those sex chromosomes. It has also been demonstrated that there are certain genetic diseases in human beings which are triggered by faulty genes which are located on the X chromosome. Such human genetic disorders include hemophilia. I hope you know what is meant by the biomedical condition called hemophilia. In case you do not know, take note of the fact that the hemophilia is characteristically an inherited bleeding disorder in which the blood of the persons involved does not clot properly. This situation can give rise to spontaneous bleeding and bleeding due to or following injuries or surgical operations. Did you know, through the principles of Mendelian genetics, pedigree analysis, and some statistical tools, scientists, including human geneticists, were able to establish the connection between many human diseases or pathologies and certain specific genes on certain chromosomes? It was this great achievement which triggered strong interest in the genetics arena on the need to decode precisely how the expression of the faulty genes do lead to the various worrying diseases in the human being. Due to the effort of various scientists across the globe, we now know with certainty that certain genes on chromosomes do control the synthesis of proteins in the cytoplasm of a cell by means of an RNA intermediate. It is the inactivity or activity of these proteins which results in the observed phenotypes among human beings. Ladies and gentlemen, friends of Daniel Hayford, thank you very much for participating in this lecture. Yes, I am truly grateful that you participated in this lecture. This lecture is accompanied by questions which will help you to assess your learning or whether you really grasp the overview which has been presented in this. So watch out for the questions and answer session with Dr. Hayford. It will truly help facilitate your learning. See you in another lecture. Bye-bye.